Hello, everyone, and welcome to today's webinar, 7 Steps for Statistical Success in Clinical Trials, Good Clinical Practice Guidelines, Part 1. Before I start, I would like to acknowledge the country. I would like to show my respect and acknowledge the traditional custodians of the land, of elders past and present, on which this meeting takes place. Before we get started, I would like to go over a few items so you know how to participate in today's event. You have joined the presentation listening using your computer's speaker system by default. If you prefer to join over the telephone, select telephone in the audio pane and the dial-in information will be displayed. You will have the opportunity to submit text questions to today's presenter by typing your questions in the questions pane of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session at the end of today's presentation. If your questions are unanswered due to time limitations, we will upload answers to these on the ACTA website. Today's webinar is being recorded and you will receive a follow-up email within 24 to 48 hours with a link to view the recording. I would now like to introduce our presenter. Welcome, James. James Matcham has over 30 years of experience as a statistician in the pharmaceutical industry. He has led the development of early clinical trial design, decisions and analysis, and the development of regulatory and reimbursement approvals for biotechnology products, involving company representation at regulatory submissions in the US and the EU. James has been a member of the Society for Pharmaceutical Statistics, PSI, for over 30 years, serving on the scientific and training committees, as well as the board of directors. He has also served on the Professional Affairs Committee of the Royal Statistical Society, RSS, where he has been a chartered statistician for over 25 years and is currently a member of the Scientific Committee of the European Federation of Pharmaceutical Statistics. In 2020, James moved to Strategic Consulting Group in Citel. Welcome, James, and I'll hand over to you now. Thank you.
Okay, thank you very much, Anisha, and thank you for the warm welcome. As Anisha said, my name is James Matcham, and I'd just like to thank the Australian Clinical Trials Alliance for the invitation to come and talk to you in the first of 2 sessions on how to become more successful as a statistician, and in particular, by improving your knowledge of good clinical practice guidelines as you go about designing, conducting, and analysing your clinical trials. So thank you for the invitation. Here's a normal disclaimer. I am an employee of Citel Inc. That's the website if you want to know more about what Citel do. The views I'm going to express are my views and not necessarily the official position of Citel. So the objective of the 2 sessions is to introduce, refresh, or update your knowledge of ICH and other international guidances as you go about your work on the planning, conduct, and analysis of clinical trials. In doing this, you need to be aware of many guidance documents, I'm sure, to ensure that you're providing very high quality work to international standards. And this is a bit of a refresher for those. So the agenda today is we're just going to do a short introduction, which is what I'm doing now, tell you about ICH and the framework that they have. And then we're going to go through the first 5 steps. So we're going to cover these following ICH documents in turn. And then the last 2 additional guidances from other regions and adaptive trial guidance we'll do at the next session. Now, the slides I'm going to present are all going to be available to you to download after the meeting. And of course, you can look at them again as you look at the recording. But there's a lot of material here, and I don't intend to go through every minute detail of every slide. However, it's there as material for you to review after you look back at these slides. What I will do, though, is signpost where to look for further materials. And I'm quite sure that after you've started to hear about some of the contents of these documents, that you're going to want to access them and read them a bit more deeply. So there's a lot of material here, and I won't be going over every minute detail on them, although I'm quite sure we could. Okay, so let's begin with the International Council for Harmonisation. What is it? It's a body that was brought together in the 1990s, early 1990s, to achieve greater harmonization to ensure safe and effective high-quality medicines are developed. Essentially, there were three groups of bodies around the world making guidances for clinical trials. There was the US FDA, the European Medicines Agency, and the Japanese authority, and they were each writing documents to give guidance on how to do trials. And in some places, they were saying similar things, and in some places, they were saying different things. And as a pharmaceutical company, that was quite hard to follow three separate sets of guidances. So ICH was brought together to try and bring them together and harmonize the guidances that were out there. I'm going to show a few websites today, but the most important one I'm going to show in the whole 2 weeks is the one at the bottom of this slide that says www.ich.org. If you access that website, you can see all of the guidelines that have gone through ICH. And in fact, all the guidelines that I'm going to show today are there. And they also have some additional materials. So there's some of the more modern guidances actually have training and slides that are actually in the website there. And in fact, some of the materials I'm going to show you have been confidently used and reused in the slides that I'm going to put in front of you today. So many of the materials that you see here are also contained in the ich.org website, although there are some that are a bit different. Okay, so ICH is made up of a large number of experts that come together in working groups to discuss the different guidances that are going to be needed over time. And they form together in working groups to go through different steps in development of these guidelines. So in May 2019, this was the number of experts, quite a large number of experts represented from different countries around the world. I did note at the top there at about 1 o'clock, there are 7 people from Australia that are actually part of ICH. I did try and hunt down who those people were, but I couldn't find out the information. Perhaps you'll be more successful than me in finding out who those people are. So moving on, the guidances are split into 4 categories. The biggest category is quality, so that's quality of medicines. There are the S guidelines, which are to do with safety. There are the E guidelines, which are to do with efficacy in its broadest sense, and they're the ones that we are talking mostly about today. And then we have multidisciplinary guidelines, which are the M guidelines. And there are a large number of these guidelines, and I'll show you some more about the E guidelines as we go forward. So here are the steps in how a guideline comes together. It starts off with identifying the need for a guidance and consensus building, all the way through to writing a technical document, drafts going out for review, regulatory consultation, adoption, and implementation. And so these are the steps that most of them go through. Probably takes 4 to 5 years for any one guidance to go through all these steps. So they do take a bit of time, but they are good value when they arrive. So let's talk more about the E guidelines. Here's quite a big table that tells you all of the E guidelines that are currently there, and also the dates that they were last issued or revised. So some of the guidelines are issued, and many of them were issued in the early mid-90s, and have been subsequently revised for use. So the ones that we're going to talk about today are these ones. So we're going to talk about good clinical practice first, E6. We're going to talk about E9, statistical principles for clinical trials. And I'm also going to talk about a revision to that one to do with Estimands. We're then going to talk briefly about E10. It's not a very big guidance, but it's important enough to include. And then just say a few words about multi-regional clinical trials. There are a number of other E documents on there that may be useful for you. Dose response studies, E4 for instance, E8, general considerations for clinical trials, safety data collection. And in the next call, we'll talk about what might be the contents of E20, which is the new ICH guideline on adaptive clinical trials. So these are the e-guidances that we're going to get through today. So let's begin with E6, good clinical practice. So here is the E6 document. R2 stands for the second revision that's happened there. And you'll notice it's an integrated addendum to ICH E6, and this is generally called GCP, the GCP document, because it's the guideline for good clinical practice. There are a number of other good practices. So there's a good manufacturing practice. And in fact, E9, before it was written, was called GSP, good statistical practice, and it ended up being E9. But the common parlance for this one is GCP, good clinical practice. In the pharmaceutical industry, when we're doing trials, we are trained on GCP at least once a year, and the training session is 2 or 3 hours to go through all of the details on this document. However, we don't have that much time today, so I'm clearly not going to go through all of this in great detail. But what I will do is just say a few words about principles, responsibilities of the sponsor, and just say a few words about protocols and amendments. This is quite a long document, and if you are interested in any of the other sections, then please get into the document, give it a good read. One of the more interesting parts of GCP is actually the glossary. It contains a really good definition of terms that I think is useful in all clinical trials. So let's take a look at principles. This is an extremely busy slide, and I'm not going to go through all of those. But in most of the e-documents at the beginning, there is a principles statement, and these are actually quite good to read. So this talks about embedding ethical principles, accepting that there are benefits of the drug that justify the risks that you're going to put the patients through. And this is to do with protecting the rights of the trial subjects, and that is quite a large part of what GCP is about. Also in here, it sets out IRB and ethics approval. And the most important one on here is the fact that the responsibility of the qualified physician is paramount. And number 8, I'm just going to draw your attention to number 8, because for the first time when E6 came out, it was stated that each individual involved in conducting a trial should be qualified by education, training, and experience to perform his or her respective tasks. And this has been interpreted very widely as being qualified by education and training and experience, and that anyone working on trials should be able to prove their education and training and experience by producing a training record for audit by any of the regulatory authorities. So these are the initial principles of E6, and I'll leave you to read through those at another time. And then more recently, there's been an addendum to E6, which actually just goes a little bit further and talks more about protection of the data in how it's accurately reported, interpreted, and verified, irrespective of the media that's being used. And this is to do with electronic records and so on. Number 11 is very important. It's about the confidentiality of records that could identify subjects. And in Europe, we had the data protection guidance that's now come in, that is now part of this. The last part then is systems with procedures that assure the quality of every aspect of the trial should be implemented. Systems with procedures means standard operating procedures, and so that means documentation of procedures and proof that people have been trained in them. So the sponsor responsibility section looks a bit like this, and you'll see me using some of the numbers if you want to refer back to sections. I'm not going to go through all of those. I'm just going to focus in on a couple. So the first one is trial design, and this is that the sponsor should utilize qualified individuals, and again it mentions specifically in trial design biostatisticians, that's good that that's there, but also clinical pharmacologists and physicians should be used throughout all stages of the trial process. And again, supervision of the conduct of the trial and to handle the data, to verify the data, conduct the statistical analyses and prepare the trial reports. So here it's talking about sponsor responsibility to provide suitably qualified and experienced people for these tasks. This one is around systems, and so this is around trial management, data handling, and record keeping. And there's a bit of guidance in here about electronic data handling, and that says that this system should be validated for completeness, accuracy, and reliability. and should be validated. And validated means documented. So there should be SOPs, audit trails of data changes, access should be limited, there should be a backup system, and there should be a safeguard to blinding, and the ability to compare to source is important. But also record keeping is also important. And there's a phrase here called essential documents, and that refers to a specific list of essential documents that also appears in the documentation. So not only is it around the people that are important here, but also about the systems too. E6 talks about the clinical trial protocol and talks about how it should be written, the format, the template, and the processes that it goes through. And again, there's an awful lot of detail here that you can get into. This is the format for the clinical trial protocol. And actually, this section does provide some discussion about what should be within each of the sections. So I just want to just pull out a couple of aspects here. One is around the trial design part. So this gives an example of the kind of discussion that the document gives around the trial design. And here are the 9 points that it goes through to say what should be in any protocol under the trial design section. So if you're writing a protocol, it's actually quite good to get the E6 document out just to remind yourself about what should be in the protocol. But there are actually two other resources that I use routinely here in the bottom right. So the SPIRIT statement is a collaboration that's come together to write guidance about the contents of protocols and standardizing protocol content. So if you go to www.spirit-statement.org, then that provides templates for protocols and discussions of contents and standards and so on. But there's also a pharmaceutical group called Transcelerate. So tranceleratebiopharminc.com. And if you go there, that actually provides templates for protocols and statistical analysis plans, actually. So that does actually provide a bit of resource here for using how to get to protocols. Just the last thing I'm going to say about E6 is about the statistics section. The guidance in the statistics section of the protocol looks a bit like this. It says that a description of the statistical method should be used. The sample size should be justified, including level of significance and so on. So you can tell that this is written in the frequentist era before Bayesian analyses came along. If you are looking at a Bayesian protocol, you have to look at the equivalent of these things. And in particular, it highlights criteria for termination of the trial, procedures for accounting for missing, unused, and spurious data, procedures for any deviations, and the selection of subjects to be included in the analyses. So it can get quite specific around those. So that's our whistle-stop tour of E6. And what I've done is to provide just an overview of the contents and some of the style of the contents for this document. As Anisha mentioned at the beginning, I'm hoping there will be 5 to 10 minutes at the end for questions. So if you do have questions, please write them onto the chat and they'll be saved up for later, and we can address those questions at the end. So the next ICH document I'm going to run through is E9, which is the biostatistical procedures. And here it is, the ICH harmonized tripartite guidelines. And it's quite interesting, it says tripartite. Remember at the beginning, the three parties were the US FDA, the European Medicines Agency, and the Japanese authority. And here then is the outline of the sections. So this document was drafted in the early 90s and has actually stood the test of time. This version comes from 1998, so you can tell how long it took to get this done. But actually it's turned out to stand the test of time, and many of the contents are still very useful. In fact, the revision to this document was only done last year. So this has stood the test of time and is actually quite a good read for anyone beginning to do clinical trials and to act as the statistician involved in a trial. Or sometimes it's actually good just to refresh yourself about what's in this document. So I'm just going to go through some aspects of this document. I'm going to talk about some general themes that run through it and get down to some quite interesting specifics. And again, we could stop and give a 1-hour presentation on any of these items. So I don't really have the time to do that today. But what I do have time to do is just to signpost the contents, what's in them, and where to look for additional help and assistance. So in the section of overall clinical development, there's a really nice discussion about primary and secondary variables. And for each of these headings, there's actually quite a lot more material in the document that explains about the roles of each of the different variables and actually does give definitions. So it talks about primary and secondary variables and the roles of the primary variable and the secondary variable. We will call them endpoints now, primary endpoint and secondary endpoint. But it also talks through the different types of variables and the things that we have to look out for as we're specifying those in our protocol. So composite variables, so time to 1, 2, or 3 things happening is an example. Global assessment variables, so overall physicians' opinion of efficacy of the drug was a very popular assessment variable at the outset, not so much now, but they did used to exist. Multiple primary variables, so this would be using more than one primary endpoint. So we would now call those co-primary endpoints. And understanding whether all of them needed to be significant or just one of them. And if it was just one of them, addressing the type 1 error if required. It also gives definitions of surrogate variables and what it takes to be confident in a surrogate variable. And also talks about categorized variables, which is usually an endpoint which is measured on a continuous scale that's dichotomized for use. And so clinical response endpoints is an example of those. So this is actually quite a good section to get into to try and understand the kind of endpoint that you have and if there's any specific guidance about using those that is available in the document. The document talks quite a lot about design techniques to avoid bias. And the cornerstone of the blinded randomized trial is stated quite roundly in this part of the document. And it talks about definitions of different types of blinding, the requirement for documenting processes against inappropriate dissemination of treatment codes, breaking the blind for an individual, and the need for that. It talks about unblinding a study, and in particular unplanned unblinding, and how to document all of that. And it gives advice and guidance about randomization. And I've just copied some of the items on here to give you a flavor of the content. So, randomized should be reproducible and secure. So you've got to keep that randomization seed. Talks about stratification factors being taken account of the analysis. Emergency access to treatment codes being provided, and about dynamic or covariate balanced allocation. And that if that's used, it cannot be deterministic. So these are the kinds of guidances that you'll see under design techniques. We are going to talk about E17, which is multi-regional trials, a little bit later. But even in E9, it started to give guidance about the use of multi-center trials. And it talked here about the design and analysis of these trials. And even talked about how to stratify the randomization by center, although that's not routinely done now, it was then. And then in the analysis about how to look for consistency of treatment effects among centers and regions. So again, some guidance here about multi-center trials. Moving on, types of comparison, and I think now we would talk about types of objective of the trial. And here in one place, for the first time back in the 90s, it talked about the differences between superiority trials, equivalence trials, non-inferiority trials, and dose-response trials, and understanding the requirements for each type of objective that might be had there. And again, there is further details on non-inferiority. If you look into some of the regional guidances around non-inferiority margins, for instance, you can see here that what was contained in E9 simply wasn't enough for definition of non-inferiority trials. And so other guidances had to be written to talk about more specifically what was required for non-inferiority trials and equivalence trials. And more recently, this has been very important in the development of biosimilar products, as most of their trials, if they have to demonstrate clinical equivalence, end up being clinical equivalence trials. So moving on, of course, there's quite a big section that talks about, let's see if I can forward my slide, that talks about sample size. A stats guidance wouldn't be right if it didn't talk about sample size. But it talks here about the aim of sample sizing and what to put in the protocol. And so quite specifically, it says those things need to be in the protocol. In essence, it's saying that what needs to be in the protocol is enough information for someone else to reproduce the sample size that you've come up with. And I think that that's, in essence, what this is saying about the protocol. It also talks about to investigate sensitivity of sample size to assumptions. And for the first time, it talked about sample size re-estimation being possible. This was even in the the early 90s that this was going on. Interim analyses and early stopping. It defined for the first time that there are planned and unplanned versions, and that there are many different reasons for wanting to do this. That most of the interim analyses should be planned, and it even gave guidance that if you have an unplanned interim analysis, the best thing you can do is plan it and write a protocol amendment and update the protocol and then do the analysis. And was actually quite specific in saying unplanned interim analyses should be avoided. So that caught everyone, didn't catch them out, but it made it quite clear that unplanned interim analyses should be avoided. The guidance talked about independent DMCs and about how there should be a written charter for data monitoring committees, including communication of results, and also about record keeping for those meetings. And again, these have stood the test of time. So even though there are some additional guidances around, this initial guidance actually was pretty good in that it hasn't really been changed since then. Pre-specification of analysis was something that for the first time in this document was seen, and meant that the protocol now had to pre-specify the confirmatory analysis that was going to take place for the primary variable to address the primary objective. And instead of just saying appropriate statistical methods will be used in the protocol, you had to say quite a lot more. And in fact, not only do you have to say what's happening about the primary endpoint and the key secondary endpoints in the protocol, it also defined the need for a separate statistical analysis plan that gave all the detail that you would need to conduct the analysis of all the endpoints in the trial. And then talked a bit about processes for the analysis plan as well. And talked about it was possible to update it in a blinded review just prior to breaking the blind. You'd have to prove it, of course. Only results envisaged in the protocol and the statistical analysis plan can be regarded by regulatory bodies as confirmatory, and that everything else was post hoc. And so it was good to write a good document to pre-specify the analysis. Again, for the first time, it brought out the concept of analysis sets. So the full analysis set and the per protocol analysis set came about on this document. And this talks about intent to treat principle, and the role of different analysis sets is actually a very good discussion in the document. This is one part that has been revised in the Estimands framework, and again, I'll talk about that shortly. But this was one of the parts that has actually stood the test of time up to a few years ago, when we started talking about the Estimands framework, and to be quite a lot more specific about exactly what the analysis is that's going to be confirmatory. So in this document, there was also an emphasis on estimation confidence intervals as well. So instead of just seeing the p-value, it's actually more important to see an estimation of the effect size and its uncertainty around that. It talked about the protocol should be specifying the hypotheses, the treatment effects to be estimated, the methods to accomplish this, and any adjustment for baseline. And again, not quite enough in E9 to be able to do this, and so the revision took place. The Estimands framework does address some more of this. What was clear, though, that in the protocol, the primary analysis should be clearly identified. So in that way, you can pre-specify the primary analysis that you're going to use to address the objective in the trial. Okay, the other one that was interesting here was around two-siding tested is preferred, but if you're going to use one-sided testing, you can just use half the significance level. So that was quite an interesting one for people. Multiplicity is also another aspect of the guidance, and it talked about the different ways that multiplicity can arise, and some of the methods for reducing multiplicity. Subsequent to this document, and more recently, there's been some really excellent guidances from FDA and EMA on multiplicity, and again, I'll be talking about some of those in the next session that we do together. Methods for multiplicity are actually required in the protocol, and this was one of the first times that we saw some of that being said officially. In E9, this is the last thing I'm going to talk about the first part of E9. There's also a section on data integrity and software validation. And again, for the first time, it talked about the credibility of results being dependent on the quality and validity of the methods and software. Used not just for the analysis, but also for the data management, data entry, storage, verification, correction, and retrieval. And so E9 does talk about the ability and the fit for purposeness of the software that is used for all of these processes. And again, the word SOP comes up again here. And if you have any procedures, then you should have data management procedures. And that the computer software used in any part of the data flow should be reliable, and testing procedures should be documented. And again, on any audit that regulatory bodies do of a pharmaceutical company, apart from auditing the fact that all the people working on the trial are appropriately qualified and trained, we also have to demonstrate that the systems we use for our data process, including the analysis, are validated and documented. And in fact, there's a saying that if it's not documented, it's not validated. Or we sometimes say if it wasn't documented, it didn't happen. So this is E9 and some highlights of E9. I think you're getting the idea that I can't get into all of the details on here. Doing a thorough session on E9 would take a couple of hours to do, and I think for another time perhaps. But I think the best thing to do is to pick up the document and give it a good read, and perhaps come into small groups and discuss it together if you haven't done that already. So I'm now going to turn to the first revision of E9, and I've decided to do this in a separate session because actually it's quite a big change to how we write protocols and analysis plans and think about objectives and the analysis of clinical trials. So this document was adopted, so this is the final version at the end of 2019 in November. And there's been a lot of discussion around the Estimands framework, which is exactly what this talks about. And the requirement for an Estimands framework came about because in clinical protocols and statistical analysis plans, the full pre-specification of the objective, the endpoint, and the analysis was not given. So there was details of part of it, but not all of it. And in particular, the reason why the objective and the analysis was taking place. And that's what the Estimands framework talks about. And so this was brought together at the, just recently. And there's some great training materials on the ICH website around this. In fact, there are 2 separate training decks on this that you can get to. So please, if you're discovering this for the first time, there's plenty of materials to get to. And there are quite a lot of groups doing training on Estimands as we go ahead. So in the next few minutes, of course, I can't give you a full treatise on Estimands frameworks, but what I can do is just talk about some of the high points. So the Estimands framework looks like this. It starts off with the trial objective and the estimand. And the definition of the estimand is the thing that it's the thing you're going to estimate. So that's what the estimand is. What is it you're going to estimate? And those should be stated in the protocol. And that defines what it is that you're actually estimating in the trial. We then have to define the estimator, the thing we're going to estimate, the main estimator, and the estimate of it. And that talks about how to estimate. And then there are sensitivity analyses to that estimator. And there'll be a number of them, and that means more than one. So 2, 3, or 4 is the number that people are going to at the moment. And they're talking about here specifying all of this in the protocol and in more detail in the analysis plan. So what it does is it actually creates more things to take care of in the protocol. Because this isn't a directly statistical thing, this is actually something for the whole clinical team to review and agree on. So it's important that this is done by the full project team and is not just something that the statisticians have to go and sort out. So what is the estimand? The estimand is a group of things, a precise description of the treatment effect reflecting the clinical question posed by the objective. It summarizes at a population level what the outcomes would be in the same patients under different treatment conditions being compared. And so the estimand definition is the grouping of the following items. It's a statement of the target population that you are estimating for, the endpoint that you're going to use, a strict definition of the treatments being compared, and a statement of the population endpoint summary, so the statistic. So it's a statement of all 4 of those things. But in addition to that, you also have to state how you're going to deal with an intercurrent event. Intercurrent events we know happen in all trials. And this is where most of the analysis plans and the protocols fell over before, because how you dealt with these intercurrent events was usually left to when you saw them. But there are a number of intercurrent events that have been predefined here. One is treatment discontinuation due to adverse events. What are you going to do with that patient if they've not met the end of the trial? How are you going to include that patient in the analysis? Treatment discontinuation due to lack of efficacy is a different thing. Use of rescue medication. Treatment switching, and sometimes death is an intercurrent event. If a patient dies before reaching the end of the trial, and if that's not an endpoint, it can become an intercurrent event. And again, it's how do you deal with that? And so the grouping of these 5 things together is the estimand. And that's what the definition is the pre-definition of all of those things. The guidance is quite good in that it does help you by talking about strategies for how to deal with intercurrent events. And here's a slide from the training, so stolen with pride here. And it talks about a number of different strategies for dealing with intercurrent events. And what we're seeing in protocols now is that you have to state which strategy you're going to use. So treatment policy is essentially the endpoint in the trial, regardless of whether or when treatment switching, changed, and so on. And this is the one that's probably most like the old intent to treat approach. However, there are composite strategies. An example there is a heart attack or treatment discontinuation due to AE. So it's one thing or another thing happening. A hypothetical strategy, for instance, is something like the change in the endpoint if rescue medication is not used. And so the question then is, how would you estimate that? And that's actually relatively complicated. The principal stratum approach, which is a definition of an endpoint in a predefined subgroup of patients. So the example here is infection severity in the subpopulation will become infected despite preventative treatment. And then the while on treatment. And so the use and pre-definition of these strategies helps you deal with intercurrent events and how to specify some of those. It also informs you how you should go about estimating missing data if it occurs under any of those strategies. And there's a lot more documentation about missing data methods to be used under each of those strategies for dealing with intercurrent events. So there's quite a lot there that's new for many folks that have been working in trials for a long time. And this is a good read as we go through. So in the guidance, it talks about how to incorporate estimands in the protocol. And again, there's quite a lot of information in the document about how to include estimands in protocol writing. And if you go back to the Transcelerate web page I mentioned at the beginning, it does give there a protocol template which incorporates estimands sections and tells you where to put them and so on. It also talks about how to incorporate estimands into the statistical analysis plan and what's required in the statistical analysis plan to give a full definition of the estimands and sensitivity analyses. I think overall the thing that I was struck by the estimand framework is that it just changes the way you think when you begin planning a trial. So it makes you go through the 7 following processes, which is to really get into the therapeutic setting and the intent of the treatment that tells you about the trial objective, identifying intercurrent events right up front and dealing with them in the protocol and the analysis plan is actually something that was done usually right at the end of the trial rather than right at the beginning. So that's quite a big difference. And then discussing the strategies to address the intercurrent events, constructing the estimands, getting the team to align on the trial design, collection and methods. Definition of the estimand also affects the data collection and the conduct of the trial and how you might do that. So that is important to understand that estimands actually does influence the conduct of the trial too. Okay, so there we are. That's the ICH E9 revision 1. I'm now going to move ahead and just talk about 2 relatively small ICH documents that are nonetheless quite important. And the first one is E10, choice of control groups. So E10, the current version is dated 2000, which is a long time ago now. But actually the choice of control group documentation has actually again stood the test of time. It's not a big document. It's got 3 sections: introduction, considerations for the type of control, and choosing the control group. And the most interesting part of this and useful part is that there is a table in this document that talks about the pros and cons of different control groups. And actually there's a flow chart at the end which talks about how to decide on the control group that you would be expected to go through. So a good thing about this document, it does give advantages and disadvantages and definitions of many different types of control group, including external control, which I know is becoming much more interesting, particularly in rare diseases, when historical controls are being used more formally in a Bayesian sense. So it does give pros and cons of all of these, and is a good read for anyone that's learning about clinical trials. Here is the table I was talking about. Let me just see if I can forward my slide. So here's a table that talks about the usefulness of specific concurrent control types in various situations. So across the top, you have the type of control. Down the left, you have the trial objective. And the letters talk about whether, yes, it's going to be useful, no, it's not useful, or P, it's possible. So this talks about how the regulators think about the control group that you're proposing for your trial, which can be useful to know and understand that. Here's the flowchart I was talking about, how to choose your concurrent control for demonstrating efficacy. And again, whichever trial you're doing, it's good just to go through this to see what regulations think about your control group. And if you are not following this, then you need to usually have a good reason. So it's not saying it isn't possible, you just need to have your arguments ready for when you want to do these. Okay, so I told you that was a short one. E10 is a relatively short document. And I want to talk now, and this is the last one that I'll talk about today, about multi-regional trial guidance. You can see from the date on this one that this is November 2017, so it's relatively recent, and came about because regulators understood that multi-regional trials were now becoming quite normal in the development of new treatments for diseases. And again, the sections in here don't look like it's a big section, However, it's quite a long document and contains some really good reading and good advice about different aspects of planning, designing, and analysing multi-regional trials. And again, the glossary is a really good part of this document and gives some really good definitions of terms that are being used. And again, there's no way in the time that I have today that I could give this the full treatment that it deserves. But actually, I will just talk about some of the key features of this document. So the first one is, again, this is a document that at the beginning talks about principles and the principles of multi-regional trials. And here are the 7 principles that it talks about. Of course, multi-regional trials increase efficiency of drug development. And by having one large multi-regional trial, you can perhaps gain approval in different regions simultaneously with the same trial. It talks about identifying intrinsic and extrinsic factors of working in different regions early, and gives definitions of intrinsic and extrinsic factors that you would need to consider. It talks about that the assumption is that the treatment effect applies to each region approximately equivalently, and that you need to understand if it doesn't and to act accordingly. It talks about pre-specification of pooling of regional countries or subpopulations. It mentions that you should plan a single primary analysis approach to this, so you can't just go straight ahead and analyse it by region. Importantly, it refers back to ICH E6 GCP and talks about no matter what region you're working in, you should ensure high quality design and conduct, irrespective of the region that you're working in, and that you should gain prior acceptance of the approach to all of these with the regulatory bodies before you start. So any multi-regional trial, you should go for prospective guidance and input from regulatory bodies before you begin. As I mentioned, this is a more recent document, so it has some great training materials available. One of the slides that I thought was really useful for you to see are the things to consider around regional variability. And there's a number of things to consider about regional variability. So dose, PK differences between regions, regional differences in standard of care, how you might train staff in the different regions. The comparators might not be approved in some regions, in which case you may need to provide them. How to collect data on ethnic factors, different regulatory positions, different inclusion, exclusion criteria, and whether they can be applied equivalently to all regions. But then there are some more intricate questions, like how to allocate the sample size to different regions, and how to plan that sample size, and how should you do that. So lots of things to consider in terms of regional variability, and this list isn't an exhaustive list. If you want that, get into the document. It's really very good at looking at all the things that you need to consider about regional variability in the application of the drug to patients. There's a really good section on how to allocate sample size to the different regions. And it does talk about 5 different approaches that you might take to allocating sample size to the regions. The one is, here they go, so there's proportional allocation. So allocating patients to regions according to the proportion of the size of the region and the disease prevalence. You could go for equal estimation in each of the regions, in which case you might want to allocate equal numbers of patients to each region. Or in each region, you might want to have enough patients to demonstrate you've preserved a certain amount of the effect of the overall treatment. And if you're doing work in Japan, that this is one that clearly you need to get to, because there's quite a specific guidance around this one. Achieving local significance, sufficient number of subjects to be able to achieve a significant result within each of the region. So a significant p-value by region, that's an interesting one. And then a fixed minimum number. So there's a minimum number of subjects in any particular region. And the approach that you go to will depend a lot on the situation that you're in and the kind of guidance that you're getting. Most groups tend to go for 1 or 2. Occasionally, it's required that you have to look at 3 if you're working in particular regions. Certainly 4 is unusual, although can be seen. And a fixed minimum number is usually present in most multi-regional clinical trials. Let's turn now to the analysis of multi-regional trials. the examination of regional consistency once you have the data in your hand would sound like a statistical exercise, but actually the guidance talks about being holistic in region-to-region variability of treatment effects, and gives some quite specific guidance around whether there should be, is there a biological reason why there should be differences? It talks about internally consistent and externally consistent consistency of results within the trial and with other trials. It talks about statistical uncertainty as being only one aspect of the holistic approach, and then the clinical relevance of these. So it talks quite a lot about this space. And again, this is another good read about what you need to do at the end of the trial. And then it does talk about the examination of this and what you might need to do in a statistical sense. So evaluation of regional consistency is not a hypothesis testing question. It's a supportive or descriptive investigation about whether prior assumptions hold true. So in this situation, it assumes that you have an overall significant effect, and you are required to do descriptive summaries, graphical displays of treatment effect by region, using forest plots, for example, model-based estimation, and the test of treatment by region interaction is only one element of this, and certainly not the final element of that. So E17 then contains quite a lot more material than, again, I can ever give in a presentation like this. And in fact, I hope what I've done is to provide some overall guidance about what is contained within each of these 5 ICH guidelines. So we're almost coming up to questions. So if you've got a question, now's a good time to be typing it into the chat. What I've done is to provide an overview of the framework for ICH, the important website, ich.org, that you would need to get to follow up on any of these documents. I've given an overview of GCP, the biostatistics guidelines, control groups and multi-regional trials. And that's been the session for today. So next time, which I believe is next week, we'll be giving an overview of additional guidance that's available out there that we tend to use when we're designing international trials, and then talk quite specifically about adaptive trial guidance and about the different number of guidances that are out there and the current situation on adaptive trials. So that's the presentation for today. I'm now open to take questions. And just a reminder that these slides will be made available after the meeting so that you can download them from the ACTA site. And again, I just take a chance to thank ACTA for the invitation to come and talk to you today about good clinical practice guidelines and the first of our steps for statistical success in clinical trials. Okay, thank you very much. I'm open for questions.
Thank you, James. That was very interesting. I have a question from Julie Marsh. She's asking that a common justification for not adhering to GCP for investigator-led trials is that they are not for regulatory review. How much reliance do journal editors and peer reviewers place on GCP?
I think GCP, although it's come around in the pharmaceutical industry, and has been the cornerstone for quality in pharmaceutical clinical trials, has been there for the pharmaceutical industry for a long while. But there's also a lot of good sense there. And it's turned into not just good guidelines for pharmaceutical trials, but actually for any trials. And a lot of the concepts that are given in the good clinical practice guidelines are actually good practice generally for all trials. So if you look at the SPIRIT guidelines, the CONSORT guidelines, and the other collaborative guidelines like that, they all look very much like the guidelines that I've been showing you today. So although investigator-led trials may not be audited in the same extent that pharmaceutical trials are done, I still think it represents good practice for any trials. In terms of publishing trials, if you've been doing your trials according to these guidelines, it puts you in an extremely good place for acceptance for publication. So if you look at the consort guidance for the content of clinical trial publications, if you've used the good clinical practice guidelines, and followed guidance for what should be in a protocol, pre-specification analysis, and all those details, it puts you in a really good position for acceptance of your publication. I think if you haven't been following the guidelines, that usually opens you up to some seriously major questions that you'll get from the journal editors. So I think it does open risk if you've not been following these guidelines. And the worst comment I think you can get with any trial is that there's the quality of the design, the conduct, and the analysis of the trial is not sufficient for publication. If you get that comment, I think that's a failure and completely avoidable. Thank you for the question. Any other questions? Okay, it appears that there are no other questions. Just remember that if you do have questions that you think about after reviewing the materials, please make contact and ask your question, and we'll try to write questions, answers to questions subsequent to the meeting.
Thank you very much, James.
Okay. Can you hear me now?
Yes, I can hear you. Can you hear me?
Yes, I can. Yes, thank you.
Okay. We do have an evaluation for the webinar, which should show up after the webinar ends. Please take a moment to provide your valuable feedback. On behalf of the Australian Clinical Trials Alliance and our presenter, James from CITEL, I would like to thank you for joining us today and have a good rest of your day. Enjoy your day. Thank you. Thank you, James.
Thank you. Bye-bye.
Bye-bye.